Hello students, this is molecular basis of inheritance part 3. So the living cells and the organisms, they are characterized by their remarkable ability to reproduce and transmit the characteristics from generations to generations and maintain the continuity of the inherited traits. So in 20th century, one of the landmark discovery in biology was the identification of DNA as the genetic material of all the living organisms. Okay, so the continued existence of a biological species requires its genetic material to be chemically stable. It's really very important. So it should be able to express accurately and have capacity to replicate with minimum errors. This miraculous organic compound DNA shows all these features, and it provides the scope and for the changes which occur due to mutations that are required for the evolution also. But it was when the DNA was supposed to be the genetic material. Okay, but before the discovery of DNA as the genetic material, by the early 1900s, when the DNA was not supposed to be the genetic material, earlier proteins, they were supposed to be the genetic material. As the proteins, they are very large, they are complex molecules, and they store more information. Suppose they were supposed to be storing more information needed to govern the cell metabolism. Hence it was assumed that the proteins, they caused the variations observed within species to species. On the other hand, DNA in comparative to proteins, DNA was very simple, it was very small molecule, and its composition was varied from species to species. It was not supposed to be stable. So the DNA was not supposed to be the genetic material, but the protein was considered as the genetic material. But over a period of roughly 25 years after the genetics, they became convinced that the DNA and not the protein was the genetic material. So these are the few experiments which you have for your study. First is this Griffith experiment, second you have Avery McLeod McCarty experiment, and third you have Hershey and Chase experiment. So out of these three, today we are going to talk about this Griffith's experiment. So this is the Frederick Griffith. In 1928, this medical officer, Frederick Griffith, he performed experiments on Streptococcus pneumoniae. This is the bacteria which causes pneumonia in humans and all other mammals. So he used two different strains of the bacteria. One strain was virulent, it was smooth, it was pathogenic, and it was encapsulated, and hence it was supposed as, or it was termed as S type of strain. Another was non-virulent, it was rough, non-capsulated, and it was termed as R type. So let's try to understand the difference between what is S type strain and what is R type of strain. So in this slide, we are going to discuss about the difference between these two strains. First is virulent, smooth, pathogenic, and encapsulated, that is S type. Another is non-virulent, it is rough in nature, and non-capsulated, that is R type. So you are having this image. Okay, first this organism which forms a rough colony. Another organism, it forms smooth colony. So these are rough colonies and smooth colony. Rough and smooth, it's based on their surface and the edges of the whatever colonies these organisms they are forming on that media. It depends on that. So these are the two strains of Streptococcus pneumoniae. R strain, it lacks the protective capsule, as R strain is non-capsulated. So now see, this is the organism, that is Streptococcus pneumonia, which is not having any coat around, that is, it lacks a protective capsule around it, and it can get recognized because of the lack of that capsule. It can get recognized and destroyed by the host's immune system. On the other hand, this another strain is this S strain, which is virulent, which is having this capsule. Can you observe the difference between these two? This is non-capsulated, and on the other hand, this another bacterial cell is having a coat around it. Okay, so that coat or that capsule, it is made up of a polysaccharide, and it prevents, because of the presence of this capsule, such type of strains or such type of bacteria, they cannot recognize or detect it by the host's immune system, and because of that, these type of strains, they are having the ability to cause virulence, or they are pathogenic. Okay, on the other hand, this R strain, it is non-capsulated, so it can get detected by the immune system and it can get destroyed, so it is non-pathogenic or non-virulent in nature. So what Griffith actually did in his experiment, there are few steps he followed. He injected R strain bacteria to mice, and mice survived. R strain is non-virulent. As we have discussed earlier, what is R strain and what is S strain. So as R strain is non-virulent, so in the first step, it was the rough strain which he injected into live mice, and the mice died. Mice survived. Okay, so R strain is non-virulent or non-pathogenic. So it doesn't harm the mouse. Okay, so the mice, they are alive. In the second step, he injected S strain bacteria to mice, and the mice died due to pneumonia. That S strain is virulent. So smooth strain, which is S strain, so he injected another strain to live mice, and he found that the mice were dead. Okay, as S strain is virulent, so as it is pathogenic, S strain is pathogenic, so due to pneumonia, the mice died. As R strain is non-virulent, it is non-pathogenic, so the mice, they survived. What he did in the third step, he injected heat-killed strain of S bacteria to mice, and the mice surprisingly survived. So what he did, heat-killed smooth strain. He heated the S strain bacteria which were virulent, okay, which were virulent in nature, but what happened, because of the heat, they lacked their ability to cause pneumonia. And so what happened, when he injected that strain, the mice survived as they were heat killed, so they lacked their ability, they lacked their power. In the fourth step, what he did, he mixed the two strains, heat killed S bacteria, now see, heat killed S bacteria and live bacteria of R strain. This is the R strain which were live. In the heat killed, the mice survived. In the rough strain of R strain, the mice survived. When he mixed these two strains, what happened? The mice surprisingly died. So he was very surprised at what happened, why the heat killed S bacteria and the live bacteria of R strain, if both are not causing the virulence or they are non-pathogenic, then why the mice, they died. So this is a diagrammatic representation of Griffith's experiment. Let's try to understand with this. He used two Streptococcus pneumoniae, that bacteria and mice. These two are were the requirements of Griffith's experiment, and he, out of these Streptococcus pneumoniae, he used two strains. Smooth, it was capsulated. There are three types of strains in that, S1, S2 and S3. All of these three are virulent in nature and all of them are pathogenic. While another strain, talking about another strain, the strain is rough, without capsule. There are three kinds of strain, R1, R2 and R3. Out of these, all are non-virulent, non-pathogenic. In the first strain step, what he did, R2, this is R2, which is non-virulent, okay, and the mice survived. S3, see, this is S3, which is virulent, which is pathogenic. What will happen? Mice died. In the next third step, what he did, heat killed S3. See, this is S3, which is virulent or which is pathogenic in nature, but he heated. So what happened? The mice survived as this strain died because of the heating process. And in the fourth step, which is very important, he took live R strain. Now this is R strain. Now see, this is R strain. The mice survived. Heat killed S3. Now this is heat killed S3. The result is mice survived. But when he mixed these two strains, what happened? The mice surprisingly died. Okay, so the question was why it happened. What caused this heat killed strain of S to become live, or what happened that the R strain got transformed to lethal or non-virulent strain became virulent strain. So this was its question, and what he concluded. Let's have a look in the next part. So he performed those three steps, and what he concluded, he recovered the large number of live bacteria from the blood of dead mice. When he performed that experiment, at the end of that, he recovered from the dead mice tissue, the tissue when he discovered that the S bacteria or S bacterial strain was found to be present in the tissue of the dead mice. So it was very surprising for Griffith. So he noticed that harmless strain of R bacteria, a rough strain which was non-virulent, changed into a deadly S strain bacteria, that is smooth strain. So R strain get transformed or got changed to the S strain. So he proved that the change was genetic. It is because of something which was getting transferred from S strain to R strain, or whatever the change is, because of the whatever transformation is carried out inside that. He was not sure about that, what he termed that, whatever mechanism it was occurred in that, he called that as transforming principle. So R strain bacteria must have taken up from heat killed S bacteria. It was his first conclusion. Second, he termed that principle as transforming principle, as there is transformation, there is change. What was the change he observed? That something was occurred miraculously, that R bacteria got changed into deadly S strain. So R strain or rough strain, which was non-virulent, was surprisingly changed into a smooth strain which is virulent. So that transforming principle must have allowed R strain to get transformed into smooth coated bacteria and become virulent. But as you know, Griffith experiment, whatever he proved, it was not much clear. So all the scientists, they were not convinced by the Griffith's experiment. Whatever he performed, it was the base or foundation for the research, but not all biologists, they were convinced. So there were few another scientists who carried out Griffith's experiment. These are the three out of these three. The second experiment, that is Avery, MacLeod and McCarty's experiment, is the tail, you can say, of Griffith's experiment. So let's talk about that experiment next. So that's all about today's session. Thank you.